Hey guys, this is the Bio channel. I'm Yusuf, and today we have a very special guest, Dr. Murakami from Penn State. Is that correct?
Yes.
And Dr. Murakami, if you want to introduce yourself a bit, some of your research, your background, how you got to where you are.
Yeah. So let me now use the virtual background because I don't want to say living
Sure.
room. Yeah. So this is Katsuhiko Murakami. I'm a professor at Penn State University. So belongs to biochemistry and molecular biology department. So I'm studying the Penn State for almost like 70 years since 2003. So my research interest is RNA polymerase, the enzyme for synthesizing RNA using DNA as a template. Then the RNA polymerase you study in the classroom, probably you study only one type of the enzyme, but there are many different types of RNA polymerases working in your body, and also bacteria, and also archaea. Now, of course, one of the recent topic is COVID. The SARS virus RNA polymerase is also one of the good target for the drug design. Then the one of the famous example is the remdesivir, only one of the few drugs approved for the COVID, which is RNA polymerase inhibitors. So my research is mainly for basic research for understanding how this enzyme recognize promoter sequence and start transcriptions. But also, I'm very interested in to use my knowledge for drug design, such as the anti-tuberculosis drug, which is the rifampicin, also targeting RNA polymerase, because RNA polymerase is an essential enzyme for life. If you can specifically inhibit some of the pathogen RNA polymerase, but not host RNA polymerase for humans, so you can design the good drugs. Yeah, so that's what I'm doing, right? Both for the biochemist, basic science, and also some of the applied science.
So basically, is it that you went into RNA polymerase research because it's so pivotal into like drug design or whatnot?
Yeah, because so when I was a grad student, I started my career. I don't know anything about biology because I was a chemistry major. But my PI is kindly introduced me the interest of the research. Then the interest about RNA polymerase, that I started my career. Then I continue my research for basic science, and then later I realized that this is, I can apply my knowledge for the drug designs, since RNA polymerase is essential enzyme for life maintenance. So that's why I'm doing both.
Interesting. All right. Now, I kind of wanted to go into the paper a bit as well. I was wondering, so the paper seems to touch on a bunch of different concepts, but towards the end, it kind of connected, trying to like go into how DEX-A and PPGPP influences RNA polymerase in bacteria.
So the factor we study the DKSA. So please remember how it's called, not the
DKSA.
DKSA, and also PPGPP. Yes.
Yeah. So is it? So I was wondering, how do you think this paper could influence future studies as well?
So if you think about the transcriptions, and then probably in the classroom, what you learn about is that DNA synthesize, RNA polymerase synthesize mRNA. Then this mRNA will go to ribosome for protein productions. Right? But if you look at the reality in the bacterial cells, for example, the majority of the RNA polymerase is also synthesizing for ribosome. The ribosome is a protein RNA complex, but the major part is RNA. So one of the main job for RNA polymerase in bacteria is synthesizing ribosomal RNA. Okay. But when the cell enter the stress response stage or stationary phase, so now the bacteria doesn't want to express any ribosome because they want to conserve the energy. So now suddenly the highly expressing RNA, which is the ribosomal RNA, is completely shutting down. So that's in that case the two factors: DKSA, protein factor for binding RNA polymerase, then another small molecule is PPGPP. This is the GDP analog, binds polymers together, then the shutting down transcription. But interestingly, so these factors binding to polymers specifically inhibit the transcription from ribosomal RNA, because other promoters, other gene expressions such as the stress response and those amino acid biosynthesis has to be continuous. So now our study, the structural biology work, studying RNA polymerase with DKSA and PPGPP complex, and those RNA polymerase bound on the ribosomal promoter complex review the mechanism, how these two factors specifically inhibit the transcriptions, and also how this ribosomal promoter is very active for transcription, but suddenly shutting down the transcriptions in the presence of these two factors.
So do you think that maybe now that you know the mechanism a bit more about how DKSA and PPGPP specifically inhibits ribosomal RNA transcription, do you think that this can now lead to like more drug design, more efficient?
Yeah, that's an interesting point because PPGPP is very important. The chemicals or the ligand for gene regulation in bacteria, not only E. coli, but also all other bacteria. And then some of the pathogens use PPGPP for expressing virulence factors. So that's why some of the researchers are thinking about making a PPGPP analog for drug. But one of the challenge for the drug for bacteria and also other drug for humans is that how to penetrate compounds through the membranes. Membrane is highly hydrophobic environment, but this PPGPP or some other nucleotide analog is highly negatively charged because of the binding to the polymerase through the phosphate backbone. That is a quite challenge for the drug designs because the negatively charged the compound is difficult to went through the cell membrane. But now my research for this particular study is mainly for basic science. I don't think anything about drug design for now for this.
Cool. And kind of like going into more, a little more details in the paper, I was wondering why you specifically chose cryo-EM as opposed to like, let's say, X-ray crystallography?
So I started my career for structural biologist at X-ray crystallography about 20 years ago, because that time only the X-ray crystallography can handle the RNA polymerase structural work because it's so too large to be studied by NMR. And also the 20 years ago, cryo-EM can reveal only the shape of the molecule, but not the detail. But X-ray crystallography has to crystallize materials before study. Then that's why we can only analyze very stable complex of RNA polymerase with DNA. So sometimes DNA has to be opened, synthetically opened to make a stable complex with RNA polymerase, then crystallize for, that takes several times, maybe several weeks or months. Then we can analyze certain structures. But now maybe 5 or 10 years ago, so now there is a resolution revolution in cryo-EM. So now the resolution of the cryo-EM drastically, revolutionarily increased. So now we can analyze the macromolecule at atomic resolutions. But also in cryo-EM, we don't have to crystallize complex. We just simply prepare complex, then prepare specimens, and analyze the structure. But also in cryo-EM, we can analyze multiple states of the complex. Occasionally, in this particular study, we have 2 structures from 1 samples. So there are some populations, the heterogeneity of the samples, but using the technique called the classifications or particle classifications, we can separate the particles nicely and determine the 2 structures at the same time. So that's give us more flexibility. Then finally, we can successfully determine the multiple states of the complex using cryo-EM.
And actually, on the topic of resolution of cryo-EM, I remember like maybe a few months ago or something that there's a paper mentioning that they now have atomic level resolution. So
yeah, so one of the challenge of the structural biology is how to visualize individual atoms. And particularly, one of the smallest one is hydrogens in this planet, right? Then the X-ray crystallography can see the hydrogen if you have a highly expressing high resolution crystals. But cryo-EM, we never believe this, but now it's happening. So we can see the hydrogens and the individual atoms by using cryo-EM. That is very amazing.
It's great. And so I wanted to ask also about the topic of scrunching. So I personally have never heard of scrunching before. I was wondering, like, what's its significance, and how does it play a role in the research you're
Yeah,
doing?
so this ribosomal promoter, there are very several interesting features compared to other promoters. These features are important for highly expressing gene in log phase, the good growth condition of the bacteria, but suddenly downregulated in the presence of DKSA and PPGPP. But one of the feature is called scrunching, which the scrunching is DNA is suddenly make shorter the length in the after making open complex. Now you can imagine that you have a spring. So if you just push the spring, so now the scrunch, and then this scrunch actually store the energy. So when polymerase start the transcriptions, so now this stored energy can be used for pushing the polymerase to the forward, moving forward. So that is the important the function of this ribosomal RNA for highly expressing genes in the bacterial cells. And then one of the interesting fact is that if you think about mRNA transcriptions, the single mRNA can produce multiple proteins after multiple ribosome binds to the mRNA, and the multiple protein can synthesize, right? But in case of the ribosomal RNA synthesis, just one ribosomal RNA can make only one ribosome, right? Then the how many ribosomes you need before cell division? Then the interesting number is that there's 72,000 ribosomes you need for making a new cells after cell divisions. Then the bacteria, E. coli, divide the cell every 20 minutes. So that's, if I do the back of my envelope calculations, every 1.7 seconds, RNA polymerase binds promoter, start the transcriptions. So that's highly expressing gene. So that's means the promoter has to be very strong for attracting polymerase for making a complex, but at the same time, polymerase can escape from the promoter rapidly. So that's why this promoter uses scrunching mechanisms for pushing polymerase going forward.
So it's almost like accelerates transcription.
Yes. So the promoter has to be strong for binding, but also moving polymerase going forward. So some of the promoter is very strong, polymerase binds, but cannot move. This is not good. But the ribosomal promoter, as I told you, every 1.7 seconds, polymerase can go from the promoter. So that's why it needs scrunching mechanisms.
All right. And then another question I had when reading the paper is kind of like the note. This is more of a notation question, but I noticed like there were different subunits with sigma, like sigma 1.1, sigma 1.2, and I have a question about that later. But is that part of the sigma 70 complex, or is that something else? In
Yeah, so RNA polymerase is a multi-subunit proteins. Then the coenzyme, which is the main part of the polymerase for synthesizing RNA using DNA template, have alpha-2. So there are the two alpha subunits, the beta and beta prime, and also omega subunits. So these 5 proteins, usually we call the subunits, bind together for making a coenzyme. But when the polymerase recognizes promoter sequence, the enzyme need additional proteins, which is a sigma. Okay? But there's a variety of the sigmas available in bacterial cells. E. coli have 7 sigma factors. Then one of the sigma factor for housekeeping gene expression is called the sigma 70. Then among the sigma 70, there is several parts of the proteins. So now this area is called domains. So they are in the same protein, but making the globular individual folding the part of the protein. So now it's called the domains. Then depending on the sequence and also depending on how they fold, we researcher like us put a unique name for individual domains. So as you mentioned, there's one of the domains, region 1.1, sigma 1.1, sigma 2, 3, and 4. So these are the name of the domains, not the subunits.
Yeah, that clears it up a lot because I was thinking about that. And actually, I found it interesting, like, because we always, it's like commonly known that, okay, so the sigma factors in bacteria bind RNA polymerase, and then you can recognize the sequence. But I never knew that, for example, there's ejection of certain domains, like sigma 1.1 was.
Yeah, because the sigma function of the sigma is for promoter recognitions, right? But once polymerase recognize promoter, then it has to change the functions, which is for RNA synthesis. And the part of the sigma binds to the coenzyme important for RNA exit and also RNA synthesis. So that's why sigma has to be removed from the coenzyme when polymerase start the transcriptions.
All right. And I had another question. I don't want to take up too much of your time as well. So I just have maybe 2 questions left. But and I was wondering, for example, the RNA polymerase to promoter interaction. And you had noted in the paper that when you substituted aspartate 256 with alanine, the interaction with G7 was more stable. Is that correct? More stable than...
Yes, that's right.
Yes.
Yeah, so this is one of the requirements for the structural biology in general. So the structural biology can reveal how the complex looks like or how the macromolecule looks like. But now that we have to, then based on the structure, we can identify some of the interactions. It looks like important between RNA polymerase with DNA. But now the time to evaluate the observation from the structure. So then one of the straightforward method is introduce mutations in proteins. So if you identify aspartic acid for interacting with particular DNA base, let's change this to alanine. Then see it functions. Now, if you see some the effect, meaning that this interaction is important. So if you don't see any effect, maybe this is not important interactions, just simply contacting, but that's it. So the structural biology can reveal interesting the interaction, but always has to be validated, evaluated using biochemical method or sometimes genetics method.
So is the reason why I was just interested that it actually stabilizes the interaction because that means like the aspartate to G7 was actually less stable.
Yeah, we saw that introducing this mutation can make a complex is most unstable, but the result was completely opposite. That we are very surprised. And still, we don't have a clear answer how it happens. Yeah, so that's the interesting part of the biochemistry and molecular biology. So you design or you have a hypothesis and design experiment. And then if you see the result in the best, following your hypothesis, you're happy. But if the result is something opposite, maybe you are disappointed. But actually, this is now time for the paradigm shift, maybe the moment. Yeah, whenever you do the experiment, you try to find something doesn't follow your hypothesis. This is the most exciting moment for you. Yeah,
that's great. And hopefully, at some point, like, is that possibly like an experiment you're looking at into understanding that?
Yeah. So now if we really want to see more detail, one of the method, the possibility is that using now this RNA polymerase mutant for cryo-EM study to see how DNA following the DNA binding pathway in this particular mutant, so that we can visually validate the effect of the mutations in the complex formations.
All right. So I think, and actually one last question, and this should be it. In terms of the DKSA and PPGPP binding, so I read in the paper that this is kind of like almost causing a global shift in the RNA polymerase protein. And so I'm just wanted to like clear this up. So is this leading to downstream effects such that DNA has to be unwound outside the protein?
Yeah, so now still this is a open question in the field. How this DKSA PPGPP influence the particular transcriptions, not all of them. But first, if you think about transcription regulations, usually the factors binds to the DNA, they interact with RNA polymerase, which influence the promoter binding, or sometimes promoter open complex formations. But in this particular case, DKSA and PPGPP do not bind any DNA, only binds polymerase. So that means the polymerase is modified, but somehow after modifications, this modification is bad for particular promoter, such as ribosomal promoter, but doesn't change anything for the other promoters. So that's a very interesting questions. But then based on the structures, we observed that after DKSA binding to the polymerase, change the conformations. And also as a published study, we now propose that maybe the polymerase use two alternative pathways from promoter binding to the open complex. Then the one of the pathway, which happens in the absence of DKSA and PPGPP, which is good for ribosomal promoter transcriptions. So the promoter can use a polymerase open channel to unwind DNA, then start the transcriptions. But in the presence of DKSA and PPGPP, so the one of the figure shows a pathway on the bottom. So now the DNA has to unwind by itself outside of the polymerase channel, then come into the channels. Then the reason why this is not good for the ribosomal promoter is that ribosomal promoter have a lot of GC-rich sequence from -10 to transcription start site, so which is the area for DNA unwinding. So now the GC have a 3 base pair and 3 hydrogen bonds between the bases compared to AT that have a 2 base and hydrogen bonds. Okay? So if you have a lot of GC sequence from -10 to transcription start site, you need more energy for DNA unwinding, or maybe the DNA cannot unwind by itself. So that's why we are now proposing that the presence of DKSA and PPGPP forcing this ribosomal promoter for the very difficult situations. Now the transcription is going down, but other promoters having a relatively AT-rich sequence from -10 to transcription start site can still work functionally.
Oh yeah, that definitely makes sense. All right. Well, I mean, thank you so much, Dr. Murakami. I don't want to take up too much time, of course. So I really appreciate you coming and giving your time to kind of explain this a bit more and kind of tell your story of research as well. So appreciate it.
Yeah.
Thank you guys so much for watching till the end. If you liked the interview, just please leave a like, subscribe, and turn on notifications as well, just so I know you guys enjoyed this type of format and so that we can have more interviews in the future. Again, maybe even leave a comment, let us know what you thought, and maybe I can forward a few questions to Dr. Murakami as well. So thank you guys so much for watching.